My name is Wendy Semsky, and I am a PhD student studying plant evolutionary ecology.
Hi everyone, I am Sumona Dhara, a doctoral candidate in the Department of Biological Sciences. So our research question mainly is to understand why some adult animals like fish can regenerate the central nervous system, while others like us, humans, cannot regenerate the central nervous system.
Hello, I am Biswarup Banerjee, an international graduate student at the Department of Biological Sciences at University of Wisconsin-Milwaukee. My research is based on molecular microbiology, and it involves the elucidation of the type 3 secretion system expression pathway.
Hi, my name is Allison, and I'm a fourth-year graduate student at UWM. I am in Dr. Erica Young's lab, where I study algae and biogeochemical cycles in Lake Michigan.
Specifically, I am interested in understanding how patterns of flower deployment influence plant mating in a native wetland plant called monkeyflower. For my study, I collected monkeyflower plants from different populations, and I brought them back to be grown at the UWM greenhouse. And then I measured how the individuals from these different populations flowered over the season. I am very fortunate to be a graduate student at UWM because our state-of-the-art greenhouse facilities allow me to conduct my experiments under very precise environmental conditions. I also conduct large-scale field experiments at the UWM field station, where I manipulate floral traits and establish experimental populations to track how pollinator foraging and behavior responds.
So to give you a brief background of what we do in Udvadia lab, here is an enlarged model of the retina, actually the eye. So you can see this is like a vertebrate eye where the retina sits at the back, and there is this portion called the optic nerve. Optic nerve is composed of long thread-like projections emanating from the neurons in the retina that wire the light information detected from the eye to the brain, where it is perceived as an image. There are two things that we expect from this study is that we can delineate an understanding of the regulation of factors driving regenerative program in fish, and this can serve as a roadmap to promote optic nerve regeneration in humans with injury or disease leading to irreversible deterioration of the optic nerve.
Our target is to elucidate and identify novel proteins in this bacterium. Our project involves a lot of molecular biology detection techniques, like measuring the green fluorescent intensity by using the fluorescence activated cell sorting machine that is present in our department, and using that we can measure the transcription of different genes. We also use the UVP machine to detect the chemiluminescence in the western blot, and we can measure the intensity of the different protein bands present in our samples. The best thing about the department is you get to learn a lot from the knowledgeable professors, and it's fantastic to work with the wonderful colleagues, and you get to learn from them every day. The best memory I have with this department is the warm welcome I got when I came to the US for the first time.
Let me take you on a little tour of what I do. So one of the things that we have here at UWM are growth chambers. All right, let's take a look inside of the chamber. This chamber is able to keep my algae in the perfect growing conditions with some grow lights, and it keeps the temperature constant at 18 degrees Celsius. One of my favorite things to do is to use the microscope, especially the scanning electron microscope, because every time I grab some sort of algal sample or bacterial sample, being able to look at it underneath the microscope, seeing all the details, it's just amazing that you can see something that's so tiny, and we can take pictures of it and see it in 3D. I have a lot of diatoms that I work with, and they make these ornate glass frustrals which have a whole bunch of micro details that you can really only see under the SEM microscope. Just, there's so much beauty that nature has made for us to see that we can only access with facilities like these at UWM.